Welcome back to Bite Size MCAT. Have you ever wondered if there's a faster way to identify enzyme classes compared to just rote memorization of each enzyme? Well, today I want to walk you through just the various enzyme classes and teach you a bunch of tips and tricks to identify enzyme classes quicker. This readily applies to both the MCAT and your various biochemistry courses, and we're going to follow a very simple if-then framework. So, to start with the basics, an enzyme is simply a protein that acts as biological catalyst that speed up reactions or perform a specific function. Think of enzymes as tiny living robots that are able to perform functions in the human body. Enzymes are found everywhere and help with functions such as digestion all the way to metabolism. And as a reminder, they do this by lowering the activation energy of reactions. They do not change the thermodynamics of a reaction, but only the kinetics. So take a look here. This is a reaction coordinate where we do not have an enzyme. So look at this tall activation energy. This is the amount of energy this reaction takes in order to proceed. But now if we add an enzyme, it decreases that activation energy, thus increasing the rate. So because there are so many different types of enzymes, the Enzyme Commission was tasked with creating a classification system in order to organize them. They created a system that has multiple classes and subclasses, but all we need to know for most applications are the first major class. So I will refer to these enzymes as EC1 or EC2, followed by their names. So let's go ahead and start going through all 7 of these classes. Our first enzyme class is an EC1 oxidoreductase. Oxidoreductase enzymes are simply enzymes that catalyze oxidation or reduction reactions, and they do this by moving electrons. So let's take a look at our example of an EC1 oxidoreductase. HMG-CoA reductase is one of these enzymes. This enzyme is part of cholesterol biosynthesis and is a great example of an oxidoreductase. Take a look at the reactant and the product. We have a change in the oxidation state, and this molecule becomes more reduced. Similarly, look at the cofactors. NADPH gets oxidized into NADP+, my apologies. If you remember back, NADPH is used for reductive biosynthesis, so this is a reduction reaction. Some other examples include oxidases, oxygenases, and peroxidases. So, if a reaction involves some sort of electron transfer, has a redox cofactor, or you can recognize that something is being oxidized or reduced, then it's an oxidoreductase. I want to spend some time talking about a special type of oxidoreductase that comes up just time and time again in biochemistry, and this is going to be the dehydrogenase. Dehydrogenases are specific in that they catalyze some sort of hydride transfer to a redox cofactor. Luckily, once we recognize this, we can think of the function of dehydrogenases. So let's take a look at an example. Alcohol dehydrogenase works by oxidizing ethanol to acetaldehyde. It does this by using NAD+ as an oxidizing cofactor, which is then reduced to NADH. As a professor once taught me, anytime you see a dehydrogenase, think of these 3 things: a hydride transfer, EC1 oxidoreductase, and a redox cofactor. And redox cofactors include FAD, NADH, NADPH, and so on. The second classification is an EC2 transferase. Simply put, a transferase transfers a functional group from one molecule to another. One of our most common transferases that we see in biochemistry is a kinase. Can you think of any kinases that we might have seen before? Well, yeah, the first step in glycolysis is hexokinase, which is a great example. Hexokinase transfers a phosphate group from ATP, which is the donor, to glucose, which is the acceptor, in order to form glucose 6-phosphate. So if a reaction moves a functional group of any kind, ends in the word transferase or ends in kinase, then it's going to be a transferase. Our third class is a hydrolase, which cleaves a bond with water. Proteases are a great example of hydrolases. A protease, such as trypsin in this example, catalyzes the breakdown of a polypeptide into smaller fragments. Trypsin is a protease that cuts after specific amino acids, such as lysine depicted here. So if a reaction uses water to simply break a bond, then it's a hydrolase. EC4 lyases are our fourth classification. Lyases catalyze the breaking of bonds without using water or ATP. So they can also catalyze the addition of small molecules such as water, ammonia, or carbon dioxide across double bonds, or the reverse elimination of these small molecules in order to form a double bond. Let's look at a classic example of a lyase, which is carbonic anhydrase, which adds water across a carbon-oxygen double bond to form carbonic acid. This enzyme is crucial for our pH buffering in our blood. Some other examples include decarboxylases, aldolase from glycolysis, or aconitase from the Krebs cycle. Synthases are a common example of lyase. So, any enzyme that has synthase in the name, think most of the time it's going to be a lyase. So, if a reaction involves a bond that is broken without water or ATP, or if a double bond is formed or broken, then it's going to be a lyase. Enzymes in our fifth class are called EC5 isomerases. Isomerases catalyze structural rearrangements between molecules by converting from one isomer to another without changing the molecular formula. Here's an example of phosphoglycerate mutase, which is the 8th step in glycolysis. It changes where the phosphate molecule is from the 3rd carbon to the 2nd carbon. Isomerases are often called isomerases or mutases, as in this example. So, if a reaction involves a molecule being rearranged without anything adding or breaking, then it's an isomerase. EC6 ligases are our second to last class. Ligases catalyze the joining of two molecules with the help of ATP. Think of ligases as ligating two molecules together or gluing them together. One example is pyruvate carboxylase, which, with the help of ATP, glues together pyruvate and carbon dioxide to form oxaloacetate. Ligases are what we call synthetases. So, similar to how if an enzyme has the ending synthase, it's probably a lyase, synthetases are probably ligases. This is a common place where students get confused. So, there are a few exceptions to these rules, but most of the time this holds. So, if a reaction involves two molecules joining together with ATP, it's a ligase. Translocases were formally made a class in 2018. These enzymes simply catalyze the movement of ions or molecules across cellular membranes. Here's going to be one of your most common examples, the sodium-potassium pump, or ATPase. This translocase catalyzes the movement of 3 sodium ions out and 2 potassium ions in using the energy from 1 ATP. So, simply, if the reaction involves a molecule or ions moving across a membrane, then it's a translocase. So there it is, a simple if-then framework for identifying enzyme classes. Understanding each of the different classes of enzymes and being able to rapidly identify them help in understanding enzyme mechanisms and how each reaction would work. So I hope this video helped, and I hope all of it made sense. Thanks for watching.